Hello students, welcome to the EPG Pathshala. I am Dr. Bhashati Banerjee from School of Biotechnology, Gautam Budh University. Here, I'll be discussing about the module DNA Replication in Prokaryotes, which is a part of the paper Genetic Engineering and Recombinant DNA Technology. So let's begin with an outline of the module. The introductory part of the module explains all about DNA, the structure of DNA, and establishes the DNA as genetic material for all organisms across all life forms. Thereafter, we attempt to understand the fundamentals of DNA replication with a general overview. DNA replication in prokaryotes is dealt with in the subsequent segment, where we focus on enzymes and components of prokaryotic DNA replication machinery. Thereafter, we move on to the detailed discussion about the various steps involved in DNA replication in prokaryotes. And also, we understand the fidelity of prokaryotic DNA replication. Let us first go through a timeline study about the DNA to understand how DNA was discovered and how DNA came to be established as the universal genetic material. Way back in 1869, Friedrich Mischer discovered DNA by obtaining or extracting it from the nuclei, and thereafter he named it as nuclein. Now by that time, a DNA versus protein debate was already hot in the circle of biological science. By that time, it was already known that the chromosome is made up partly of protein and partly of DNA. So there was a strong lobby which advocated protein to be the genetic material of all organisms, and there was a budding lobby which advocated DNA to be the genetic material of all organisms. However, the confirmation, the conclusive proof that DNA is nothing but that universal genetic material was obtained through a series of subsequent experiments. In 1928, Frederick Griffith was the first person to demonstrate the bacterial transformation. Along with this experiment, he postulated the concept of transforming principle, that entity or that object which carried the genetic information from one generation of bacteria to the subsequent generation. Since the molecular nature was not yet established, so Griffith termed this genetic carrier as the transforming principle. Thereafter, taking the experiments of Griffith one step ahead, in 1944, Oswald Avery, Colin MacLeod, and Maclean McCarthy were the ones to determine beyond any doubt that DNA is nothing but the transforming principle, with a series of experiments where they utilized several methods to individually denature the protein, denature the RNA, denature the carbohydrates, and denature the DNA in the bacterial extracts with which the mice was inoculated. There was conclusive proof and sufficient evidence that the transforming principle is made up of nothing but DNA and DNA alone, and that genes are composed of DNA, neither protein nor carbohydrates nor RNA. Thereafter, in 1952, Alfred Hershey and Martha Chase provided the final convincing evidence and conclusive proof that DNA is the genetic material. And not only genetic material, DNA is the universal genetic material, as they proved that even the phage, which is a virus, used DNA as its genome or genetic material. By early 1950s, it was already established that DNA is the genetic material for most of the organisms. Later on in 1956, Geiger and Schramm discovered that RNA happens to be the genetic material in some plant viruses such as the TMV. So it was known by now that in the course of evolution, at some point of time, both DNA and RNA happened to be the genetic material of the organisms. So what was the reason that evolution ultimately chose DNA as the universal genetic material? Let's see the reasons one by one. There is a well-accepted hypothesis that RNA was the first molecule of heredity. It evolved all the essential methods for storing and expressing genetic information before DNA came into scene. However, evolution replaced RNA with DNA as the universal genetic material. The first and foremost reason is DNA being double-stranded is more stable and resistant to damages. The double helical structure of DNA is capable to protect the nucleotide bases by keeping them inside the structure, and also the double helical structure helps in identifying any error that occurs during replication. Next, the DNA is chemically more stable compared to RNA, so it was possible to maintain greater lengths in case of DNA double strands in comparison to RNA single strands. The hydroxyl group of RNA at the 2' carbon position makes RNA extremely susceptible to hydrolytic attack, which being absent in DNA made it chemically resistant. DNA was more capable in identifying and repairing replication errors due to the presence of thymine instead of uracil, due to the fact that cytosine mutates to uracil due to deamination. Now that we know that DNA was established as the universal genetic material, what was left was to discover the structure of DNA. The first convincing step towards elucidating the structure of DNA came with Erwin Chargaff's rule of equivalence, which stated that DNA is composed of equal amount of adenine and thymine and equal amount of guanine and cytosine, the ratio being A to T is 1:1 and G to C is 1:1. Following this, in 1953, James Watson and Francis Crick proposed the highly acceptable double helix model of DNA structure by combining the critical findings of Rosalind Franklin and Linus Pauling. In that same paper, they also proposed the semi-conservative nature of DNA replication. However, this proposal, this hypothesis needed to be verified, and this landmark experiment was performed by Mendelssohn and Stahl in 1957-58. We will now go into the details of understanding of the fundamentals of DNA replication. But before going into the details of Mendelssohn and Stahl's experiment, we should first know what other models of DNA replication were suggested. Nevertheless, they were ruled out after evidences provided by Mendelssohn and Stahl in their landmark genetics experiment. Although the semi-conservative mode of replication was proposed, yet the other suggested models included the conservative replication of DNA, semi-conservative replication of DNA, and dispersive replication of DNA. In the conservative replication model, DNA replication results in one molecule that consists of both original DNA strands, identical to the original DNA molecule, and the other molecule consists of two new strands with exactly the same sequences as the original molecule. In the dispersive replication, DNA replication results in two DNA molecules which are mixtures or hybrids of parental and daughter DNA. However, the most accepted and most effective model of replication is the semi-conservative mode of DNA replication. As per this model, the two strands of DNA unwind from each other and each acts as a template for synthesis of new complementary strand. This results in two DNA molecules, each carrying one parental strand and one new daughter strand. With a path-breaking experiment of Mendelssohn and Stahl, the two other models, that is, the conservative model of replication and dispersive model of replication of DNA, were totally ruled out, and the only one DNA replication model that came to be established and universally accepted was a semi-conservative model of DNA replication. Mendelssohn and Stahl designed a classical genetic experiment where they grew the E. coli for 14 generations in heavy ammonium chloride medium, following which the same E. coli was grown in normal ammonium chloride medium, after which extracts of every generation was subjected to ultracentrifuge and the UV dosimetry was done, which showed that with each replication generation, the percentage of normal ammonium chloride kept increase. The percentage of normal nitrogen in the E. coli extracts kept increasing exponentially, and the percentage of heavy nitrogen in the E. coli extracts kept decreasing exponentially. This could be possible only if each generation contained one parental strand and one daughter strand. For the same experiment, they are also credited with inventing the density gradient centrifugation technique, which today is being used for several other genetic experiments. Let's now understand in detail the nature of DNA replication. First and foremost, the DNA replication is of semi-conservative nature. As we already discussed earlier, during replication, the parental DNA duplex unwinds and each strand then acts as a template for synthesis of a new daughter strand in antiparallel and complementary direction, such that the newly synthesized pair of DNA duplex contains one parental strand and one daughter strand. Now, in case of DNA, the two strands are present in antiparallel orientation, which means that in the DNA duplex, one strand is present in 5' 3' orientation, while the other strand is present in 3', 5' orientation. Now, significant feature of the DNA polymerase, that is the enzyme which replicates DNA, is that it can synthesize DNA only in 5', 3' direction, although it can read the template only in 3', 5' direction, which means that on one of the two strands which have unwound in the origin of replication, in one of the two strands which is present in 5', 3' orientation, the DNA replication happens to be semi-discontinuous in nature. Now, a characteristic feature of DNA duplex is that two strands are present in antiparallel complementary orientation, such that the 5' terminus, 5' phosphate end of one strand corresponds with the 3' hydroxyl terminus of the other strand. When the DNA duplex unwinds at the origin of replication, one of the two strands are present in 5', 3' orientation. The other strand is present in 3', 5' orientation. A significant feature of DNA polymerase, the enzyme which replicates DNA, happens to be that it can read the template only in 3', 5' direction and can synthesize new DNA or add nucleotides only in 5', 3' orientation. Thus, the strand which is present in 3', 5' orientation is easily available for the DNA polymerase to read and synthesize DNA. But in the other strand, which is present in 5', 3' orientation, the DNA polymerase can start reading it only after it has opened for a few nucleotides, and it synthesizes in the reverse direction compared to the direction of replication fork. As such, this process has to be repeated multiple times in the 5', 3' strand. Also, replication in the 5', 3' strand begins a little while later compared to the 3', 5' strand. Thus, the 3', 5' strand is known as the leading strand, while the 5', 3' strand is known as the lagging strand, on which the replication occurs in a discontinuous manner in the form of multiple short stretches of oligonucleotides known as the Okazaki fragments. In the 3', 5' strand, on the other hand, replication occurs in a continuous, seamless manner. Thus, overall, the nature of DNA replication is semi-discontinuous in nature. Now, the replication fork, which is formed at the site of origin or origin of replication as it is also known, It proceeds in both directions, and on either direction it is flanked by one replication fork, one replication bubble flanked by one replication fork at each end proceeds opposite to each other. Thus, replication occurs in a bidirectional pattern. Before understanding the mechanism of DNA replication in prokaryotes, we must first understand that the DNA content in prokaryotes, as we all know, is distributed in two forms. One, the genomic or chromosomal DNA, and the second, extrachromosomal or non-genomic plasmid DNA. The major similarity between the two is both of these are composed of single closed circular DNA molecule which is present either in coiled or supercoiled form. We will, however, focus only on the genomic DNA replication in this module. So, let's first understand what a genome is. Genome, as the definition says, is the haploid set of chromosomes in a gamete or microorganism or in each cell of a multicellular organism which carries one complete set of genes or genetic material as must be present in a cell or the organism. So, we will begin with discussing a few important features of prokaryotic genome. To begin with, the prokaryotic genome is composed of a single circular chromosome. It has a single point of origin of replication and only one point of termination. It is present in the cytoplasm, not enclosed in any nuclear envelope, but it remains attached to the plasma membrane at a specific point known as mesosome. The prokaryotic genome is devoid of introns and has complete exonic region, or in other words, coding genes only. Before proceeding further, let's have an overview of all the enzymes involved in prokaryotic DNA replication. First and foremost, for any kind of DNA replication, DNA polymerase is the most essential enzyme, which is the only enzyme that is capable of adding deoxyribonucleotides to the growing strand. Then comes the role of helicase, the enzyme which helps in unwinding the DNA duplex. Primase is an enzyme which is equivalent to DNA-dependent RNA polymerase. Now, what's the role of primase here? DNA polymerase, of all the known types of DNA polymerase, it has been seen that none of the DNA polymerases have de novo synthesis capacity, which means the DNA polymerase cannot start synthesizing DNA chain, which means that the DNA polymerase cannot start synthesizing DNA without a pre-existing nucleotide. However, the primase, which is DNA-dependent RNA polymerase, has the capacity of de novo synthesis. So, at the beginning of replication, primase in the form of DNA-dependent RNA polymerase synthesizes a short chain of oligonucleotides made up of ribonucleotides or RNTPs. This is known as the RNA primer, based on which the DNA polymerase extends the newly replicating DNA strand. Later on, the RNA primer is removed by DNA pol 1, which acts as an exonuclease to remove the primer. Thereafter, the newly synthesizing the nascent DNA strand is extended by the action of DNA pol 3. Other important enzymes are sliding clamp, which holds the replication complex in position, the single strand binding protein, which stabilizes the single strand of DNA, topoisomerase, which helps in unwinding and releasing the tension created by unwinding of DNA, and DNA ligase, which seals the gap between consecutive Okazaki fragments. Now, let's understand the details of DNA polymerase which are used in prokaryotic system. So far, 3 different types of DNA polymerase in prokaryotes have been known. The DNA pol 1 is the first polymerase to be discovered in prokaryotes by Arthur Kornberg, and it is actually this polymerase DNA Pol 1 which totally revolutionized and opened a new dimension in molecular biology. This is a 103 kilodalton protein having one major subunit. However, proteolytic cleavage can disperse it into two smaller fragments. The large fragment known as the clino fragment having a 5' 3' polymerase and 3' 5' proofreading exonuclease activity. The smaller subunit having only 5' 3' exonuclease activity, which helps in removal of the RNA primer post synthesis. The exact number of subunits of DNA polymerase II is not known, but it is known to be involved in DNA repair function and shows prominent 3' 5' proofreading exonuclease activity. DNA polymerase III is the largest of DNA pols. It's a holoenzyme having nearly 10 subunits, out of which the alpha, epsilon and theta subunit form the core enzymes and the other subunits form the accessory enzyme activity. It shows 5' 3' polymerase and 3' 5' proofreading exonuclease activity and acts as the main replicative polymerase in prokaryotes. In order to understand the details of prokaryotic replication, we must also focus on the origin of replication, which in case of bacteria is also known as ORIC. It has a characteristic feature which includes 3 prominent 13 nucleotide 80 rich sequences and 4 prominent 9 nucleotide sequence also rich in adenine thymine repeats. 4 distinct proteins which bind to ORIC happen to be the factor of inversion stimulation or FIS, the SIC-A, the DNA-A and integration host factor protein. Upon binding of these proteins to the ORIC marks the initiation of replication. Pre-replication complex, which assembles at the origin of replication, mainly includes DNA A protein, which binds tightly to one of the 9 base pair consensus sequence present in the ORIC. The replication in prokaryotes begins with the assembly of primosome and priming of RNA primer. The Primusome complex, which assembles at the origin of replication, includes the DNA A, DNA G primase, the DNA B helicase, DNA C, which is a helicase assistant, other proteins being DNA T, pre-A, pre-B, and pre-C. These 7 proteins combine to form a multi-protein complex known as the Primusome complex, which assembles at the bacterial origin of replication, thus triggering the unwinding, opening, and stabilizing of the DNA single strands. Priming includes synthesis of a short stretch of ribonucleotide oligonucleotides, which acts as the RNA primer, upon which the DNA polymerase is able to extend the newly synthesizing DNA strand by adding deoxyribonucleotides to the available terminal. The reason being, of all the known DNA polymerases in bacteria, none of them have a de novo synthesis ability, which however exists with the RNA polymerase or DNA dependent RNA polymerase, which in case of bacteria is the DNA G primase, which is able to synthesize the RNA primer de novo, upon which the DNA strand is thereafter extended. Let's try to understand the molecular dynamics and events which occur at the replication fork during bacterial DNA replication. As we have already discussed, the replication on one of the strand occurs in a continuous, seamless manner, which is known as the leading strand, while on the other strand, the replication occurs by formation of multiple Okazaki fragments in a discontinuous manner, and that is known as the lagging strand. During every initiation of replication, the primase synthesizes a short RNA primer, after which there is an event called polymerase switching, where DNA polymerase III takes over the activity of polymerizing, and the RNA polymerase, also known as primase, is replaced by DNA pol III. This happens in both the strands. However, the time is different. While the leading strand continues to replicate ahead of lagging strand, the priming and polymerase switching occurs multiple times in the lagging strand. The replication fork is stabilized by the Replisome Complex, which is again a multi-protein complex. This happens to be conserved across all organisms. However, the components of replication complex are different in different organisms. In case of E. coli, which is a model organism for prokaryotic replication, the replisome includes DNA pol III, clamp protein beta, the clamp loader gamma and tau complex, a single strand binding complex, a DNA G protein, and a helicase DNA B. Let's understand the stepwise events that occur during replication mechanism in prokaryotes. To begin with, the pre-replication complex assembles at the origin, which leads to opening up of DNA duplex by helicase. However, it should be noted that a single-stranded DNA is quite unstable in nature. Hence, it requires single-stranded binding proteins to coat and stabilize the single-stranded DNA, which has now opened up to form the replication bubble. One large replication bubble is formed that progresses in both directions and is flanked by one replication fork at each end. Topoisomerase is the enzyme that binds to the DNA duplex ahead of replication fork. The main function of topoisomerase is to prevent DNA strand breakage by supercoiling, by which it achieves by releasing the tension in DNA duplex. Primase is the DNA-dependent RNA polymerase that synthesizes a short stretch of RNA polymer required for synthesis of DNA strand by DNA pol III. Thereafter, DNA polymerase I starts adding nucleotide to the 3' hydroxyl end of the RNA primer. There comes the event of polymerase switching. DNA polymerase III replaces primase and DNA pol I and continues elongation of both the leading strand and the lagging strand, albeit in the form of Okazaki fragments in the lagging strand. RNA primers are removed by the exonuclease activity of DNA pol I, and the gaps between Okazaki fragments are filled up by DNA pol I by adding DNTPs. The gap between two consecutive Okazaki fragments is sealed by DNA ligase through formation of phosphodiester bonds. We will now understand in details the different steps of DNA replication. The whole event of DNA replication may be classified in 3 steps: initiation, elongation, and termination. Let's begin with understanding initiation of DNA replication in bacteria. It begins with assembly of pre-replication complex at ori C, which recruits the Primusome complex, thereby triggering the unwinding and opening up of origin of replication, forming 2 individual single strands of DNA template. These are stabilized by single-strand binding protein, which is a part of the Primusome complex. Thereafter, RNA priming begins by the activity of primase, and the initiation of DNA replication is then taken up by DNA polymerase I. Finally, the DNA polymerase III holoenzyme assembles as a part of the Replisome complex, which switches from primase and DNA pol I, and thereafter DNA synthesis continues seamlessly on the leading strand and in a discontinuous manner on the lagging strand in the form of Okazaki fragments. The next event in DNA replication is the event of elongation. Now, whenever there is elongation, it requires the formation of a replication bubble flanked by one replication fork at each end. And whenever there is a replication fork, there must be an assembly of Replisome Complex. The components of Replisome Complex we have already seen earlier. Here we discuss about the bidirectional progression of replication fork and replication bubble. A dimer of DNA polymerase III holoenzyme elongates both the leading and the lagging strand always and invariably in the 5' 3' direction, while in the leading strand the elongation continues seamlessly, but in the lagging strand elongation continues by formation of multiple Okazaki fragments in the 5' 3' direction. The DNA polymerase III alpha subunit polymerizes DNA while the epsilon subunits do the proofreading on both the strands. The genome of bacteria happens to be composed of a single circular chromosome where the point of origin of replication and the point of termination lie nearly opposite to each other at 180 degrees of respect to each other. We have already discussed in detail the characteristic features of ORIC. Now let's understand the characteristic features of the ter elements which form the termination region. The ter elements are 23 base pair sequences which form the sites for binding of TUS gene protein. These are arranged on the chromosome in two clusters with opposite orientations so that these create a trap for the replication fork. The replication fork may only enter once in these regions, but it can never escape out. Thus, it is the middle of the ter sequences where the two replication forks meet and the replication terminates by releasing of the replication bubble and subsequently releasing of the two new chromosomes formed. Since DNA replication is a pivotal process for survival of an organism, it must be done with extreme accuracy and precision. Nevertheless, the rate must not be compromised to increase fidelity, or the fidelity must not be compromised to maintain the rate. Now, fidelity of DNA replication is different for different organisms, and it can be measured or expressed in a number of ways. The bacterial system, as we all know, is a prokaryotic system which is evolutionarily much lower organism. However, in spite of the fact that it is a very primitive creature, the replication machinery, that is the DNA replication mechanism, on one hand it is highly conserved, on the other hand it shows high fidelity of DNA replication. The bacterial DNA polymerase replicates at a very rapid rate by adding nearly 1,000 nucleotides per second. Yet, in spite of this high speed and also in spite of the fact that it's a very primitive creature, isn't it really interesting to know that the error rate of DNA replication is as low as only 10 to the power 9 to 10 to the power 11 base pairs replicated. This high fidelity is achieved by three distinct steps known as the base selection, which gives a 10 to the power minus 4 to 10 to the power minus 5 of error-free rate, followed by 3', 5' exonuclease, that is proofreading exonuclease activity, which edits, giving an escape rate of nearly 10 to the power minus 2. Then comes the mismatch repair step, which includes removal of remaining mismatches, giving an escape rate of nearly 10 to the power minus 3. So let us summarize what we have learned in this module. First, we understood that accurate copying of genetic information in the double helix of DNA is essential for inheritance of traits that define the phenotype of cells and the organism. The core machineries that copy DNA are conserved in all three domains of life: bacteria, archaea, and eukaryotes. In the prokaryotes, which are argued as the original replicators, DNA replication involves more than 30 specialized and essential but conserved proteins. Thus, DNA replication in E. coli, which is one of the most well-studied replication processes, provides a model for studying fundamentals of DNA replication. This machinery includes many conserved elements, which hence may be explored as potential targets for drug targeting and provide a platform for designing novel antimicrobial therapeutic strategies. We wrap up the module here. Thank you.